Hi everyone, my name is Tanya Irani. I currently go to Wilfrid Laurier University and I'm under the supervision of Dr. Moreno Hegelsib. And today I'm going to be talking about my research, which focuses on considering the taxonomic classification of E. coli. So classifying species has always been this gray area in science, whether it be in terms of animals, plants, or bacteria. And what causes this grayness is the fact that there's so many different species concepts that can be used to classify different strains, different animals, and different plant species. So methods of classification for bacteria have also been evolving through decades. And over here, what I've done is create a little timeline of classification. So starting off, we have phenotypic testing, which looks at things like the morphology of the bacteria, as well as doing some biochemical testing. So for example, with E. coli, we test for things like lactose intolerance. So this is also used as a first step today in terms of bacterial classification. So the problem with this method, however, is that it is all experimental and there's a lot of room for discrepancy, especially when you look at things like the way that a bacteria looks. So in the 1960s, we were introduced to DNA-DNA hybridization. So what this is that it compares strains by the way that the DNA of the two strains anneal. So if you had one unknown strain and you had a strain that you knew was a part of the species of E. coli, you could extract the DNA from both, and then you'd see if it annealed at a rate of 70%. And if they did, then both those strains belong to the same species. The only problem with this is that, again, it is an experimental method, so that leaves a lot of room for error as well. So it was also a very slow method. So they were looking for something that was also a little quicker, because to classify tons and tons of genomes would take forever because of how slow it is to do this. So in the 80s, I believe, it was 16S RNA analysis was introduced. So what that does is that it looks at a small portion of the genome. So it just looks at the 16S RNAs of those genomes, and it was actually used before DNA-DNA hybridization. So it would be used as like this initial step to classify these clades, and then you'd go in and then do DNA-DNA hybridization on those individual clades to further classify the strains. So although this is a much quicker method, it's also not as accurate because you're only looking at a small portion of that DNA. So then we started a very common technique that's still used today, is looking at the average nucleotide identity. So what this means is that you're comparing full genomes with each other. So you have to align them and you have to compare strains and to see if they have a 95.5% similarity, because that'll tell you if they belong to the same species or not. So the only problem with this is that it is, however, a very time-consuming method. So obviously not as time-consuming as DNA-DNA hybridization, but it does still take a long time. So a new method that is trying to be implemented is looking at the DNA signatures. So what exactly is a DNA signature? So a DNA signature is actually the observed over the predicted values of a di, tri, or tetranucleotide. So for example, over here, to look at the DNA signatures of AA, for this species in particular, we look at the observed dinucleotides of AA, so that's 0.7, and then the predicted, which is 0.6 in this case. So for example, over here, our DNA signature for AA would be 1.17. So now how can we use this for a method of classification? Well, in work done by Dr. Moreno Hegelsib, what he did was that he found that DNA signatures of strains of the same species actually condensed at a distance of 0.03. When working with E. coli, though, however, he found that they broke off into 3 different groups, meaning that they condensed at a much higher distance. So this suggested that E. coli strains actually have genomes that are different enough that they should be considered as different species. So 3 different species in this case. And the reason that he was trying to implement this method, once again, is because of the efficiency of it. So for example, when comparing the average nucleotide identity and the DNA signatures, it was found that DNA signatures only took a week to determine for all the DNA signatures of 584 strains of E. coli, versus the average nucleotide identity, which took 1 month to find. So the work that I'm doing currently actually stems off of my undergraduate thesis project. So what I did then was look at 3 different methods of classification for E. coli. So I looked at the average nucleotide identity, the 16S RNAs, and the DNA signatures. So for my average nucleotide identity, I found the average nucleotide identity for 584 strains of E. coli, which was the amount of E. coli strains last year in the NCBI database, and I clustered them at a similarity of 95.5%. For my 16S RNAs, I determined all the RNA identities, and then I clustered it at a similarity of 98.5%. And then for my DNA signatures, I determined those again for all the strains, and then clustered it at a distance of 0.03. Now what this gave me was what I was expecting, that all three methods were going to give me different results. So for my DNA signatures, I got 3 clusters, once again, with the biggest containing 324 strains, and with the smallest, very interesting, 26 strains. The average nucleotide identity method gave me 560 strains of the 584 that were considered as the true strains of E. coli. And then my 16S RNA method gave me 542 strains, which was in the biggest cluster, the true strains of E. coli. And then there was 27 other clusters. So the reason, first of all, to note why my 16S RNA, there's so many strains is because there's multiple 16S RNA identities for each genome. So that was not unique. So that's really important to note as well. And then also another thing that I found interesting was between my average nucleotide identities and my DNA signatures, the average nucleotide identities had the 24 strains that clustered themselves out. That was found in 9 different clusters. Those 24 strains were all the same strains that were found in cluster 3 of my DNA signatures method, giving me the idea that maybe I could use the DNA signatures in combination with my average nucleotide identity as a better method of classification. But that's something I'm going to get to. So based on the results of all 3 methods, if I were to create a cluster just on that, I would find that there would only be 317 true strains of E. coli as compared to the 584 that there actually were in the database last year. Another interesting thing to note is that in the NCBI database, there's actually strains of Shigella and Citrobacter that are found in the species of E. coli. So their genomes are similar enough that they should be considered as the same species. So when I did this reclassification, I actually expected those strains of Shigella and Citrobacter to be the ones that group themselves out. But what I found was actually the opposite. I found that the strains that were labeled as Shigella and Citrobacter actually had genomes similar enough that they should be considered as part of E. coli. And the strains that grouped themselves out were all labeled as E. coli. So those genomes were actually different enough that they should either be considered either a subset of the species of E. coli or a complete species altogether. So my main takeaway were these 3 things from that project that I did. One, that there was a lot of discrepancy between all the methods, which is what I was expecting, but the thing that they all had in common was that it showed that E. coli was not classified correctly. The third thing that I took away from it was that DNA signatures actually has a possibility of being used as a better method of classification. So it gave me some hope there that I would take forward to the project that I'm working on currently, that I could figure out a way to make implement DNA signatures as a better method of classification when used in conjunction with something else, probably. So the 2 main questions I was looking at, my first one was what makes each cluster of E. coli unique? So that's what I really wanted to determine, whether those differences, they would lie in strains in parts of the genome that are part of the core genome, or second, the parts of the genome that were horizontally gene transferred. And then the second thing, the main question that I'm looking at is how can DNA signatures be used to better define species? So originally I thought it was something to do with the threshold that DNA signatures was being used at, but I'll get to that. I did take a look at that. And then now I'm thinking that it's probably that I can implement it by using it in conjunction with something else, just like how 16S RNAs were used in conjunction with DNA signatures for a more efficient method of classification. So the 3 objectives that will be guiding my project are, 1, to build a hierarchical cluster for all 1,072 strains of E. coli. So currently in the NCBI database, as it was updated, there's actually 1,072 strains of E. coli as compared to the 584 that there were last year. So that's almost like a full double in the amount of strains that there are. My second objective is to determine what parts of the genome are causing the strains to be classified into different groups. So as I mentioned before, whether they're genes that are horizontally gene transferred or they're genes that are found as part of the core genome. My third objective is to then determine a way that DNA signatures can be implemented to classify species accurately and efficiently. So this is really my driving objective that I'm working towards. So what I did first was that I actually created trees based on 5 different methods of classification that I'll be focusing on. So I created trees based on the average nucleotide identity and the genomic similarity score, as well as the 16 sRNAs, the 23 sRNAs, and the DNA signatures. So I put trees that kind of looked more similar to each other together right next to each other. So I'll be getting to the other 3 trees as well. So I'm still working on kind of determining how the E.coli are grouped exactly. So over here, although these 2 trees are similar to each other, there's still so many differences, and that's really what I wanted to highlight, how every single method gives a different result in terms of classification. So over here I have my 16 sRNAs and the 23 sRNAs. Obviously we have more 23 sRNAs than 16 sRNAs, which is exactly what was expected because there are more 23S RNAs for each genome as compared to the 16S. And then I have my DNA signatures. So over here, this is actually a tree based on DNA signatures for all species, but I'm still working on determining where the E. coli is in this tree. So also for my DNA signatures, before I got into the rest of it, what I wanted to do was make sure that the threshold that the DNA signatures was being used at was the best that it could be being used at. So I took a look at the di, tri, and tetranucleotides for thresholds between 0.01 and 0.05. So the 0.01 is a lot more conservative as compared to the 0.05. So the reason I stopped at 0.05 is because all 3, the di, tri, and tetranucleotide signatures, the amount of clusters it gave was only 1 in that case, meaning that it was too liberal over here. Now 0.01 I can tell is much too conserved because I have 58 clusters of the tetranucleotides and 20 clusters of the trinucleotides. So the main ones that I've been using is the trinucleotides. And I did figure out that the signature distance it was being used at was perfect. It wasn't too conserved and it wasn't too liberal as well. So the main methods that I focused on, so again, I did analysis of the 1,072 genomes, so I'm still working on analyzing all my results. So I classified them based on the 5 different methods. So that included the DNA signatures, the average nucleotide identity, the 16 sRNAs, the 23 sRNAs, and the genomic similarity score. I then now want to determine where the differences are in these genomes. So I want to analyze all the different methods and all the different clusters by looking at things like the GC content, the genome size, the number of proteins, the genomic islands, and the functionality of the proteins. What these things are going to tell me then is whether these differences will lie in genes that are part of the core genome or genes that have been horizontally gene transferred. So for example, if I find that some clusters actually have signature genomic islands, that means that those genes were horizontally gene transferred, which means that though that cluster would be actually considered a subset of the species of E. coli as compared to a different species altogether. So if differences lie in the core genome, I know that that actually causes them to be a part of a different species altogether. So I then want to determine a more efficient strategy for classification. So I want to figure out the way that DNA signatures can be used better for classification. So this will be based on using it in conjunction with something else because I've already taken a look at the DNA signature thresholds and I've determined that it is being used at the best threshold right now. So I want to figure out what other method I can use it in conjunction with. So the way I'll do this is possibly by using DNA signatures first to identify clades because it seems to be a little bit more, it is much more liberal than the average nucleotide identity. But if I can figure out the clades quickly using the DNA signatures method, then it would be a lot more efficient to use the average nucleotide identity to identify strains in those clades. So that is what I will be looking at next. And then from there, I want to create a new tree for E. coli. So using this improved strategy that I will determine, I want to then figure out the improved groups and clusters for E. coli. So that would wrap up kind of my whole project. So what is the significance of all this? Why do we care so much about classifying species? So it's really important to be able to classify species because we want to be able to better understand strains that can't be cultured in the lab. And also this project is going to work on ways that we can more efficiently classify different strains and species, because it is really important to have some efficiency as well as accuracy. So yes, thank you so much to everyone in my lab as well for all the support throughout this project. And that is my contact information up there. Thank you.